Hi, this is a Science Hangout from Science Mission. I'm Sadashiva Pai. We have today Dr. Indira Mysorekar from Washington University School of Medicine with us. She is Associate Professor in the Department of Obstetrics, Gynecology, Pathology, and Immunology. Dr. Mysorekar was born in India, where she lived for the first 8 years of her life before moving to East Africa with her family. She lived there until she was 15 and then moved to US for her school studies. She did her undergrad and masters in Sweden and again moved back to US for her graduate school. So currently she is the associate professor in Washington University School of Medicine. She is the recipient of multiple awards, including a prestigious 5-year pathway to independence from NIH. She holds the Investigator Award in Reproductive Sciences at Burroughs Wellcome Fund. She has also received National Research Service Award from National Cancer Institute. In addition, she also won a Postdoctoral Fellowship Award and Academic Women Network Leadership Award. Let's introduce Dr. Mysorekar. Welcome, Dr. Mysorekar.
Thank you very much, Sadashiva. It's very nice to be here this morning.
Thanks for joining us. It's really pleasure to have you here.
My pleasure.
With us. Can you talk about some of your background, education, etc.?
Yeah, as you mentioned, I have lived in many different countries and grown up in different countries, which has been interesting because it's different to do science and education and different cultures. But I've benefited a lot. I've learned a lot from the way things are done or perhaps not done in Tanzania, and the way science happens in Sweden, and the pace at which it goes, and then to be here in the United States at a very competitive institution, it's a different ballgame. But having survived in different areas makes you stronger in some ways.
That's good. So you have a pretty good research career, and you have more ahead of you.
I hope so.
So can you please let us know about your some of the people who helped you, some of the role models, mentors you had in your career?
Yeah, definitely. I mean, my my greatest and my guru, as I would say in India, is my father. My father was also an academic like me. And when I was growing up, I wanted to be just like my father, and it makes me very happy to have followed his path. And one of the reasons I wanted to do that was because he not only was a great educator, a great scientist, but he loved all aspects of science. He loved, of course, the research, the intellectual challenges of going into a new area and discovering new things. My father, when he was doing his early studies, he was one of the first in colonial India to go to Oxford University in England, and was one of the earliest people studying the nascent field of isotope geochemistry and nuclear geology. So he discovered some of the early radio dating of rocks and so on. So it's very exciting to do that. The second thing he liked was the collegiality of scientists, because no matter which country you go to, before you go to a lab or you speak with a scientist, you have a common language. So he had friends everywhere in the world, and all of them, we visited them, they visited us. And so I grew up in feeling that all world, all science was all the same. You know, it was wonderful. I felt safe and comfortable as long as there was science and scientists around. And he loved educating and mentoring, and he was a great guru. And his earliest PhD students are now in leadership positions around the world. And even he just passed away a few months ago. And thank you. And by that time, he had 4 generations of trainees and mentees. who still stayed in touch with him and would come visit him and honored him and so on. And so his legacy lives on infinitely beyond his specific life, and that's something that I admired and wanted to emulate myself. And so I'm still on that path. But so he was my greatest role model. He's the one who encouraged me. And then beyond that, it's been great scientists. One of the earliest people that I admired was James Watson, the double helix, and Francis Crick. That made me want to study molecular biology, which is what I majored in college. And since then, it's been more people in my field, people who have trained me and people from whom I've learned. One of the things that has been in common when I look back in my life, I've been doing research since I was 15. I spent every single summer of my life doing research, and every single lab I've worked in has been a very large lab, and I like that. I like different model organisms. I like taking a systems biology approach to my science, and even when I set up my own lab, and I was small, it was a small lab in the beginning, we still took the same approach, which was a little challenging without the resources, but that's just the way I think, and it's worked out really nicely in my career because I've been able to be in many different areas and contribute to many different areas, and always stay excited about something or the other because something's always working.
Okay, so in addition to science, I think you are doing other things. Your family, you are spending time with the family.
Certainly.
Can you talk to us something about your hobbies and what else do you do other than working?
Oh yeah. I do a lot of things. I love. I'm married and have my husband's also in the same field, different area, but the same kind of job, I guess. And we have a son who's 14. And I love gardening. We have a beautiful garden that I've built essentially from scratch because we bought our house, it was all ivy. And now it's pretty much all year round garden that blooms from February through November. And we cook a lot, and I love hosting dinners where I do multiple course dinners with different with a lot of fusion cuisine, and invite people from different places, just like my parents did, have people visiting us from all over the place, and I love traveling. So, which is perfect for scientists because we travel anyway, and I love that, and I have friends everywhere and like to go visit. I mean, I have other interests too, but those are the main ones.
Okay, and talk to us about your lab. So how it is organized, lot of people and all.
Yeah, so I was very fortunate when I first started my lab here, because we get what's called a startup package when we set up our lab, which essentially pays for part of our salary and allows for money to buy equipment and reagents and so on. But along with that, as you mentioned, I got the Pathway to Independence Award, which is a five-year award that was almost $1 million, which was really great because that was on top of my startup. So I was able to start on a very strong note and hired a lot of people early on, which is unusual for a starting lab because usually you start with one tech and then you build from there. But I hired people and really invested in all my money in people and developed them. So that's allowed my lab, I think, to be very strong and grow bigger. Work in multiple areas much more quickly than if I had done a traditional start. The other, and so then it led to, and plus, even though I'm a basic scientist, I've always been very interested in translational research, which basically means that I like to do things that can go from the lab to the patient pretty quickly. And so I have been very involved in speaking with the clinicians in my department, and each of the areas in the department have fellowships, which are essentially their postdoctoral fellows. So these are people who have finished residency and are now in their fellowship, and usually they do research in their fellowship years. So we have set it up so that people who are either studying, who are either doing high-risk pregnancy or urogynecology fellows come to my lab.
Yeah.
And then medical students come to my lab as well. So we have a very nice mix of senior scientists, senior postdoctoral fellows, but also a number of PhD students, MD PhD students, and clinical fellows, and then a series of undergraduates. And because as I mentioned, I started studying, working in a lab since I was 15, and I was always very grateful to all the people who gave me an opportunity, because I didn't know anything, but they gave me an opportunity anyway. And so I swore that if I ever had my own lab, that I would nurture young scientists. And so I've had high school students and undergrads, and now I've trained about 14 of them. And I'm extremely proud. These are my children too. So.
Okay. And talk to us about your various research projects and the work you are currently today presenting.
Sure. So I'm going to present one of an area that's extremely hot right now and much in the news, which is the Zika virus epidemic, especially in pregnancy. But the reason we came to that, which has only been is less than a year since we've been doing it, and but I came to it from I first started for my PhD research in studying host microbial interactions in the intestinal epithelium, in the intestine. And so that was my first foray into looking at interactions between commensal microbes in the gut, our gut microbiome, with the gut epithelium and the gut mucosal immune system. So I was studying what my advisor used to call the trialogue between the immune system, the microbes, and the mucosal lining. And so I got very excited about that. I like looking at things holistically and looking at all the interactions as opposed to a deep signaling pathway. And then when I did my postdoc, I moved into another mucosa. I started studying the urinary tract, where there is there's not like a rich microbiome associated with it, but the urinary tract is susceptible to very common infections called urinary tract infections, or UTIs, which are particularly common in women. So I started studying that area and made two major discoveries during my postdoc. One discovery was that these E. coli that caused infections established reservoirs inside the bladder mucosa, and when, even though that people seem recovered and there's no active infection, clinically you might have been treated by antibiotics and you don't have any bacteria in your urine, there's still a very high risk of recurrence. And recurrent UTIs are particularly bad because often they become refractory to antibiotics. So then you get more and more severe infections. And so when I discovered that there were these reservoirs, that was a big breakthrough, that the bladder itself could be seeding new infections. And those reservoirs are not visible to antibiotic therapy, because antibiotics are only in the bloodstream and the mucosa, and they don't go intracellular. So identifying that was huge. And so when I set up my own lab, now I've continued that has led to a project trying to identify first, how do the bacteria get to this reservoir? What is this reservoir? What is this location? And how do the bacteria survive long term? So that's been one area. And in that process, we've discovered that a recycling system called autophagy is essential. Autophagy is a recycling mechanism that is present in every cell. And the main purpose is to recycle dying mitochondria and misfolded proteins, anything that needs to be degraded, so that new amino acids are produced and then they go back into the cycle. It's just a recycle cleanup system. And it's also important for recycling or killing microbes, which are foreign objects that are coming into the cell. But we discovered that E. coli, uropathogenic E. coli, which is the bacteria that cause urinary tract infections, are able to subvert this mechanism and use it. So they actually go into a structure called an autophagosome, which is a protective double membrane structure, and persist there for long periods of time. And then we asked the question, how is it that the bacteria are living in this little vacuolar space? Because it's not nutrient rich. So how are they able to do that? UPAC bacteria are not obligate anaerobes. They typically don't have an intercellular life cycle. So how are they surviving? So that led us into a new area, which we're very interested in, which is iron biology. So turns out, of course, we know iron is an essential element. It's essential for all of our normal functions and everything. And both our mucosal cells and immune cells need iron for normal functioning. But so, which is why these cells store iron, but no one stores free iron, which is what is needed for the functions. So iron is stored in complex with a substrate called ferritin. And whenever the cell needs iron, this ferritin-bound iron complex is processed through autophagy and cleaved so that free iron is released and then can be used. So it turns out that this process of ferritinophagy goes into all the ferritin and iron goes into the autophagosome, and then it's cleaved and free iron is released. So the bacteria are constantly getting food coming to them, which is basically the reason they can persist. So now we're able to starve the bacteria. Now we have an avenue to get at the bacteria without antibiotics, is that we can block ferritinophagy, we can chelate iron, or we can inhibit autophagy. We've done this in cells, we've done this in animals, and our next step is now to translate it into humans, where if you can modulate dietary iron so that the person is not, or the animal is not anemic, but you chelate enough iron so that the bacteria loses the battle and they get starved out and then they don't come back out again. So this would be a non-antibiotic method of getting at those bacteria. So that's one area. And then the second area, which then comes back, this is still in the bladder, is that when the E. coli bacteria first come into the mucosa, they invade into the bladder cells and multiply and lead to a lot of death of these cells, a lot of death and damage. And a lot of the cells which are important for barrier function in the bladder, because you see the bladder stores urine, which is a waste product. So you don't want any waste products in the urine crossing from the urinary space back into your bloodstream. So to do that, the mucosal lining is very impermeable. But when you get an infection, a lot of the impermeability is lost because the cells are dead, because they've been infected. And so in order to not compromise our host physiology, there's a very extremely rapid stem cell proliferative program that gets activated to replenish these barrier cells. So it's one of the most remarkable things that the bladder normally would only turn over once in 6 months. But when you have an injury, like this infection, it starts turning over within 6 to 24 hours. It's one of the most rapidly orchestrated stem cell regenerative response ever known in all mammalian systems. So I was very excited about that, and I started identifying the mechanisms behind this stem cell activation, and ended up identifying a series of pathways that were really important for this purpose. And then going back to my original interest of looking at the mucosa, the bacteria, and the immune cells, turns out that certain immune cells are extremely important in modulating not only the immune response to bacteria, but they're equally important for orchestrating the regenerative response after the infection. So you have to kind of modulate it very carefully, because you need the immune cells to produce pro-inflammatory cytokines to get at the bacteria, but then you want them to switch and it starts to creating pro-regenerative molecules to help with regeneration. So we started looking at all the mechanisms involved in that. And that led us into looking at aging, because in aging women, in postmenopausal women, so basically women over 50 or so, who have low levels of estrogen, this entire process is significantly delayed, this regenerative process, and the immune response process. And because of that, so the barrier doesn't get restored. So postmenopausal women are at incredibly high risk of getting multiple infections. And so we started studying that, and now we've been able to, because we understand the mechanism, immune mechanism, the regenerative mechanism, the mechanism of bacterial persistence and recurrence, we're able to put all those together, integrate it to study the pathogenesis of UTIs in older women. Well, in aged mice, but since we have our Gynecology colleagues and fellows in the lab, we have samples and from women who come to our clinic, and we have a large clinical study where we're getting patients. And now we're trying to integrate both of our immune research as well as dietary iron research, both in our mice and in our women, concurrently. So we're extremely excited about that. So that's the bladder, and then when I started my new job here in this department as a new assistant professor in 2008, there was a lot of interest in my bringing things that I had developed in the urinary tract to see if it was also happening in infections in the placenta during pregnancy. So it's been well known for a long period of time that many women go into labor early and deliver prematurely, and it's a pretty big burden on society to have a premature baby, because often it's more prone to infections, its lungs are not as well developed, and many other things. They can be at risk for a number of conditions later on. And infections have been most strongly linked with preterm birth. And so any woman who's at risk is often put on prophylactic antibiotics or given antibiotics to reduce the risk of infections. But when they did a large randomized control trial where they gave prophylactic antibiotics to some pregnant women and not to others, they found that the women who were given antibiotics, in fact, had a larger risk of delivering prematurely than the women who were not, which completely threw a loop, threw a loop down, because now people are like, well, what do we do now, And so when I came in, they wanted me to investigate. We started to investigate whether there were reservoirs of bacteria, just like we found in the bladder, that may be causing these infections, which would also be not amenable to antibiotic treatment. And so we started looking for these microbes, and we found them. But we found them in pretty much every placenta. So then at first we thought we were doing something wrong. And it took us a couple of years to sort of figure out that no, it was not wrong. Turns out that the placenta has its own microbiome, and which it's always been considered sterile. And so whenever people saw any microbes, they were treated with antibiotics, which it wasn't necessarily the best thing. So it turns out the placenta has a microbiome. So then we published the first paper that showed that there were microbiota that might be living normally in the placenta. And then that started us thinking, so we're investigating the placental microbiome. And then that also started us thinking, so if there's some bacteria that are normal and not infectious, that means that there must be mechanisms in place in the placenta to differentiate between the two. And also, the placenta is remarkably resistant to most infections, because think of all the things that we might be faced with, but most babies are still born normally without being infected. So we started looking at not so much the infections in the placenta, but the microbiome and normal defenses. What are the normal host defenses in place? And that brought us again to autophagy. And so there's an area in the placenta where fetal cells are bathed in maternal blood. This is where all the nutrient exchange happens. And so that area has very high levels of autophagy. And because anything that's not good that comes in is recycled immediately. So there's just high basal level of autophagic activity. And turns out that if you modify this autophagic activity level, you now suddenly increase the risk of getting an infection. So that is so we're now studying autophagy in the placenta, which has been also extremely exciting. And in the process of all this, the Zika epidemic came on, and one of the biggest aspects of this infection, the reason it came into the news so much was because babies were being born with these small heads, this condition called microcephaly, where it's like much smaller than normal. And so suggesting that something was happening in pregnancy that was unusual. So we immediately started investigating what was going on, and we were the first to show. We had a paper in Cell in May of last year showing that the Zika virus, even if the mom is infected subcutaneously, like through the mosquito which carries the virus goes and crosses the placenta, and in fact overcomes this placental barrier and is able to cross over into the fetal blood vessels and fetal bloodstream, from where it goes into the fetal brain and kills a lot of cortical neuronal progenitors, which is what in large part leads to the small brain structure forming. The cortex simply doesn't form. And so we identified that there was a transplacental route. And then most recently, we had a paper in Nature last month, which was very exciting, which showed the first therapeutic intervention, which was a monoclonal antibody against the viral envelope protein that actually could block the virus from crossing the placenta. So it stopped it on the mom's side, did not let the virus cross to the baby side, and the fetuses were born normal and undifferentiated from, indistinguishable from wild type pups. So that's been huge. And so now we're continuing to look at placental barriers and how does Zika basically break the barrier, and using Zika now kind of as a method to understand how the barrier can be broken. So as you can see, I work on a lot of different areas, but they're linked by the fact that we're just chasing the question. And if the question led us to a new methodology, then we adopt a new methodology, and or we collaborate with someone who has that methodology. So I just always believe in following the scientific question. Go where the science leads you.
And you have the data to share with us today?
Yes, I'm very happy to share the data. Are there any questions that I can answer?
I think after the data presentation, I'll ask a few questions.
Okay, perfect. All right. So this is just a few slides highlighting. Can you see the slides?
Not yet, no. Have you shared it on the?
Oh, that's right. I have to do that. 1 second. Are you able to see it now?
Yeah.
Okay, great. So can you see my cursor? Okay, yep. Perfect. So I've just put together a few slides. This is all recently published data, and I just wanted to give highlights. So if you have questions, I can go into more detail later. Hold on 1 second. Okay, so a lot of this work, of course, it's always the people who do the work. And this work has been done tirelessly by a most brilliant postdoc, Dr. Bin Sao. He's been in my lab now 5 years, and he is ready to take off on his own soon. He's interviewing for faculty positions. So by the end of the year, he should have his own lab, and he's just been extraordinary working literally working day and night in this very fast moving area. So can you see the whole slide?
Yeah.
Okay, perfect. So as I mentioned, Zika virus is typically transmitted via mosquito. It shares a lot of homology with the West Nile virus, dengue virus, yellow fever virus, a lot of these viruses that are tick-borne or mosquito-borne. And it's a single-stranded RNA virus. Basically, it doesn't have protein. So here's the RNA structure, and these are the proteins. This is the non-structural proteins that are important for the envelope. And one of our colleagues solved the crystal structure last year. And so not much has been known about this virus. It was discovered in 1947 in the Zika forest in Uganda, which is what gave it its name, Zika. And they knew that it could infect monkeys. And that was all that was known. There was a small paper from 1947 describing that. And then there have been sporadic episodes here and there in the world, and which led to the understanding that that mostly if you get infected, you have these symptoms like fever, there can be a skin rash, joint ache, headache, and conjunctivitis. But typically symptoms go away between 3 to 5 days. There was an epidemic, a small epidemic in French Polynesia in 2007, which brought Zika to attention for one of the earlier times, because people infected with this virus had Guillain-Barré syndrome, which basically is like temporary paralysis in adults. And so they had many cases of that, but the biggest interest started coming in 2015, late 2015, when suddenly there was a lot of talk and news coming out of Brazil, especially the northern part of Brazil, in the Amazon forest area, in the summer, that there were babies being born with small heads, and thousands of them. So it was like just like that. Suddenly there was thousands of babies. The hospitals were getting flooded with them, and so then they immediately started investigating and realized that they were all infected with this one virus, which was identified to be the Zika virus. The other troubling things that people saw was that not only was the head small, but there was what's called intrauterine growth retardation, which is basically a small baby. Even if it didn't have a small head, the whole body was small. It was just small for its size. And there was another thing called placental insufficiency, because the placenta that basically nurtures and feeds the baby when it's in utero was also smaller. So everything was smaller. The placenta was smaller, the baby is smaller, and so on. And so now we know in humans also that there's in utero transmission from mom to baby. And now we also know that there's sexual transmission. So it can go from male to female and also from female to male. But male to female, of course, is the risky part for pregnancy, because the virus could survive in the vaginal mucosa, and if it's in the vagina, it can go up and infect the pregnancy and infect the baby. And now we also know that males can get infection of their testes, and the virus can persist in the testes and semen for a very long period of time as well. So it's a pretty bad virus. Anyway, so given these things that were coming out about the small babies and with small brains, we wanted to immediately start working on generating animal models to study the pathogenesis and figure out how this infection was happening. And we focused on mother-baby transmission and trying to prevent that in an animal model. So to do this, we collaborated with Mike Diamond, who's a famous and very well-established expert, flavivirus expert. So he's worked on West Nile, dengue, and all these. He's a very, very well-known viral immunologist here at Washington University. And he has had an interest in new viral epidemics, emerging viruses for a long time. So he heard about Zika at a meeting in 2015 on emerging viruses. And so he decided to start developing animal models to be able to look for this study this infection. The problem with this infection is that it's a human infection. So the virus actually doesn't necessarily cause infection in a wild type mouse. So he ran through hundreds of mouse models to identify mice that could get infected. And he identified mice that were deficient in something called interferon receptor alpha and interferon alpha beta signal, which is important because that is a mechanism that the virus uses to, it has to inhibit interferon signaling in order to do its infection. And that's why a lot of antivirals are interferon activators, because that's what gets rid of most of these viruses. So anyway, so so then where we got in with him was to study pregnancy. So we basically took interferon deficient moms, so female mice, and we mated them with wild type mice so that the placenta and fetuses would be heterozygous. Okay, so we took those crosses and infected them, the moms, subcutaneously to mimic a mosquito bite with the Zika virus at different days during pregnancy. And then we analyze what happened afterwards. And basically, you can see here, this is a wild type this is a wild type animal infected with the virus. These are all the different fetuses with their own placenta. It's all looking normal. Whereas the IFNAR het moms, their placentas are all black and the fetuses are resorbed. And even the ones that you could see, the fetuses were much smaller throughout, and the placenta is smaller, and a lot of these pups didn't get born. Then this is in the IFNAR knockout mice, but he also developed another model where we could take wild type mice and block them with an antibody against interferon receptor signal. So this you could use in wild type mice and still be able to block it. And when we did this, it was a slightly milder phenotype, so we could look at the pups that were getting born, and we could see that if you had the infected IFNAR het mouse, this is the level of resorption. So most of the pups died. Only a few were born, and those that were born were very small. But if you treated with the anti-IFNAR antibody, you had some of the pups that were born, but they still had the small bodies and the placenta were smaller, which is what is shown on this slide. So we examined the placenta from these animals, and this is the myometrium. This is the muscular lining of the uterus. This is part of the mom's uterus, and then this area is called the decidua, which is the former uterus that becomes part of the mom's section of the placenta. Here's the fetal placenta, and then you have the umbilical cord going into the fetus. Okay, so the baby would be on this side. So this is a wild type, and this is in the knockout, just mock infected, they look normal. And this is in the IFNAR mutant mouse. You can see the placenta is much thinner and smaller. And when you look histologically, here's a normal histological section, some blood cells, some trophoblast cells, and then in IFNAR het infected, you see there's a lot of loss of cellularity and these collapsed blood vessels. Then when we looked deeper at the trophoblasts, so just to orient you for those of you who don't study the placenta very much, here again is the myometrium, which is the muscle layer. Here's the decidua, which was formerly the mom's uterus, that becomes the decidua and offers the outer layer of the placenta. Then this is in the mouse. Then there's the junctional zone and the labyrinth. This is the junctional zone, which has cells called the trophoblasts, which perform a lot of the functions of the placenta. And then you have the labyrinth. All of this area here is mother's blood, and these are fetal. This is red is fetal blood. Let's see here, and around it are the fetal capillaries, fetal endothelial cells, and in between are additional trophoblasts which form the barrier. Okay, so these trophoblasts are shown here, and the nuclei are trophoblast nuclei. These are placental cells, and all the red are viruses. So if the virus can go into the trophoblast, it multiplies and essentially damages the trophoblast. So the virus comes in from the spinal artery, goes into these trophoblasts, and basically destroys its way through, and then goes into the fetal endothelial cells. I'll go through this pretty quickly, but here is a virus here that's replicating and getting assembled in the rough endoplasmic reticulum of the fetal capillary. So you can see that the virus is actually like making more of itself in the fetal capillary. And here is a stain in red for the fetal capillaries. So here are the nuclei, and here are capillaries. These are normal capillaries in an uninfected animal, uninfected placenta. And here is an infected placenta. You see all the capillaries are collapsed because they've been infected and damaged by Zika infection. Because once it goes from here all the way across the trophoblast layers where it kills the trophoblast, goes to the fetal endothelial cells, kills them, collapses them, and then it's in the fetal blood. And from there, it can go straight into the fetal brain. And this is a review that we published in the New England Journal of Medicine last summer, which basically summarizes our findings, which here's the virus. It goes into these maternal arteries from the mom's blood, goes in, and it crosses the junctional zone, and then goes into what's called the labyrinth zone here. And this is where it crosses from the mom's blood across the trophoblast into the fetal capillary, and from there it can go into the brain. So this was pretty clear evidence that the virus was taking a transplacental route to the baby. And so then, of course, so knowing that, we want to be able to stop it. And for this, one of the major efforts we've been taking is to try to develop new vaccines or antivirals. And for this, we had a collaboration with Jim Crow, who's chief of the Human Vaccine Center at Vanderbilt University. And he basically gets blood samples from the CDC from anybody who's been infected with Zika, and he isolates any human monoclonal antibodies from that infected blood, and then screens the antibodies against binding to an envelope protein of the virus, and then identified one monoclonal that had strong activity against the Zika protein. And then we put this virus into our mouse model of pregnancy. And basically, the results were really fantastic. So again, here's the placental structure. Here are animals that were treated before infection and also after infection with the monoclonal H117 antibody. Here's an uninfected animal. You can see that the monoclonal basically essentially got rid of the virus in the placenta. And here is our mice that were treated with a monoclonal against chikungunya, which doesn't do anything. And here is a Zika infected animal. So you can see there's a level of infection, and here's relative to uninfected. So overall, the placental area, the placental thickness was restored. So this is smaller placentas, this is normal placenta, and the placental area is restored. And when we looked histologically, the cellularity of the placenta was essentially restored. So we didn't have any epithelial, any damage, trophoblast damage. We looked at where the viruses were. And here again, placentas, this is the chikungunya control. This is Zika infected animals just treated only with the PBS control. And here's the H117, here's the placenta. And when you look at where the viruses are, you see them all through the placenta here and here, but here you see that there's no more viruses. So basically, the viruses just did not cross the placenta. And that resulted in a much lower placental load overall. And then finally, looking at the fetus and then endothelial cells, here's the H117 treated animals, and their vessels look as normal as those in uninfected animals, whereas the Zika infected ones still had a lot of damage of the fetal endothelial cells. And then finally, when we looked at the fetuses themselves, we found that most of the fetuses were intact, and the fetus size was pretty much the same as an uninfected animal. And there was very limited fetal load of viruses in the fetus itself. So basically it just didn't go into the baby. So now, so that was tremendously exciting. And that antiviral monoclonal is being tested in non-human primates, and soon should be going into human clinical trials. So we're extremely excited to have participated in that. So now we're looking at a variety of questions, most predominantly, what's the mechanism? Now we know it crosses the placenta, and so how does it do it? So now we're going much more detailed into the cell biology. We're also looking at, does it matter when you get infected during pregnancy? It appears that if you get infected earlier, the severity of infection is greater than if you get infected later in pregnancy. So we're investigating that. And then of course, we want to translate a lot of our findings into our human studies. So that's just a few of the highlights of our work, and happy to answer any questions.
Yeah, thank you so much.
You're welcome.
Can you unshare it?
Yes.
Yeah, it's a very neat work. I mean, like in such a short span of time.
Yeah, it's a short time, not even a year for all that.
Yeah, you are able to get some very nice publications and some good work. And can you talk a little bit more on those antibodies? Like, are they in the clinical trial at the present, or like you're planning on that? Like, how does it go?
Yeah. So that is not in my hands, because that's being done by our vaccine colleagues in Vanderbilt. But they are in the early stages of going into clinical trials. So.
Okay. And regarding the molecular mechanisms, like are you looking at the biochemistry? What is the pathway? What receptor does it bind, and all those things?
Yeah. So my colleague Dr. Diamond is investigating things from the viral perspective and the immunological perspective. We are using our understanding of placental trophoblast biology and trying to understand, like, how is it that the virus breaches the placental barrier. So that's the area of interest right now, and we're putting together a new paper on our understanding of that. So maybe next time I talk, I can share that with you. So.
Okay, that's good. And one question is regarding, like, it's like one of the major thing is the microcephaly.
Yes.
So like, and recently I think I've seen some other, like, it affects other organs also. Like,
yes.
So do you know, like, why it is more pronounced in the brain cells rather than
I think that has become pretty clear with a lot of work that's been done in this area. So basically, this virus appears to have some specific tropism. So it has tropism for placental trophoblasts, which is one of the reasons why it goes and multiplies there. It has tropism for cortical neuronal progenitors in the brain and the cortex. And so basically when it goes into the brain, it kills the progenitor cells. So when it does that, and glia and radial glia, is when it does that, you just basically don't get development of the cortex. It doesn't affect many of the other cells. It seems to affect, and it doesn't affect differentiated cells. It seems to affect the progenitors. And the same thing in the testes. It appears to affect spermatogonial stem cells, not mature sperm, but it affects the stem cells. So it's killing them. And so it seems to go to these specific places. It doesn't seem to do anything in the liver or in the spleen or even in the intestine, so clearly it's got, like, it goes at things that are very important to human reproduction and human existence. So that's why it's very scary. It's sperm, stem cells, placental cells, and brain cells,
So yeah, like, do you know, like, why specifically, like you mentioned about troposomes or whatever, but is there any why that it happens only to these organs?
Yeah, so this is a question that's been kind of confusing. So the clear hypothesis would be that there must be receptors on these cells that the virus binds to. And it's true that these cells have receptors. They belong to a tyrosine kinase receptor family. They call TAM receptors. But how they're functioning in these cells is still a little confusing because people have made mice where they knocked out these receptors and it didn't make any difference to the infection. The infection was just as bad as normal. But in some human cells, they've blocked the receptor and they see some reduced infection. So the literature is a little bit confused right now. Are these receptors important or not? Are they maybe not sufficient? Maybe there are other receptors. So I think all that stuff is ongoing right now.
Okay. And now going back to, this is the last question. I don't want to hold you up more. Regarding the UTI, you mentioned that in the, there is a reservoir of these viruses. Does it happen in all other organs? Like we have viral infections, like for example, cold and all those things. So some people keep on getting those cold quite frequently, but not others. Yeah. So do you think that similar mechanism exists in other organs?
Yeah, that's a great question. Yeah, yes and no. I guess you may have sometimes it's a question of the virus or the bacteria itself persisting long term in reservoirs, and sometimes it's a question of what kind of immune response is put in place. And if you have an active, continued chronic immune response, then it doesn't need another. That doesn't need the physical microbe. Any new microbe that you get exposed to can trigger the same response. So typically, but a lot of people don't. This is what comes down to with studying things more holistically, is that you think about all these things. Whereas many microbiologists look at them from the microbial perspective and see what happens in the microbe. A lot of immunologists look at what happens in the immune cells. A lot of physiology, host tissue people look at host damage and after infection, but nobody necessarily, a lot of people don't necessarily look at all three things at the same time. So it's possible that other infections also have reservoirs. I mean, certainly we know toxoplasma can do this in the brain. Listeria could potentially, I mean, there's definitely a malarial parasite makes reservoirs in the liver. Chlamydia can make reservoirs in the vagina and cause infections. So certainly it's known for some of them, but whether all the other infections also have reservoirs, I think has not really been looked at.
I think we are almost out of time. So again, thank you so much for taking your time and joining us. I really appreciate it. So good luck with your research, and look forward to more of these hangouts with you.
Great, me too. Thank you very much for inviting me.
Thank you so much.